Okay. Hi, everyone. Thank you for joining. Welcome to the 9th session of the International IVF Initiative. So I'm Eva Schenkman, and I will be moderating this session for you along with Tony Anderson. I'm again honored and excited to be co-hosting this session of the International IVF Initiative. For videos of previous sessions, please go to ivfmeeting.com. You'll also find information there about upcoming sessions on that page. So we are continuing our goal to support the IVF community during this COVID-19 pandemic by providing these educational opportunities and while bringing our community together, which is quite evident in all of the messages and people representing many countries from around the world. Today's program is brought to you by the CRB, the AAB College of Reproductive Biology. We have a great program planned for you today with two great talks, one on uterine microbiome and one on sperm DNA fragmentation. I3 has a talented team behind the scenes to post your questions to our speakers. Some questions will be answered live and some will be typed in the Q&A section. So I will now turn the program over to Tony Anderson, who will begin our discussion.
Thank you, Eva. It is a great opportunity to be here today to have this meeting, as a number of our meetings were canceled around the world. These were a couple of talks that we had planned for our AAB College Reproductive Biology meeting. So our first speaker today is Dr. Inmaculada Moreno, the relevance of uterine microbiome and IVF treatment. Dr. Moreno has studied biochemical sciences and molecular microbiology at the University of Valencia, Spain. And today she's coming to us from Valencia, Spain, to present her data to us on the microbiome. During her postdoctoral training, she specialized in molecular oncology and regenerative medicine. Dr. Moreno is currently the research manager and leads the research on the role of the microbiome in human reproduction, research and development department at iGenomics. Dr. Moreno has been productive in both basic and clinical research with more than 30 international publications in peer-reviewed journals and presented this data in over 50 international conferences. So I'm pleased to announce Dr. Inmaculada Moreno, the relevance of uterine microbiome in IVF treatment.
So thank you very much for this nice presentation. And thank you, the I3 initiative for having me here today. And thank you to all the participants. I see that there's a lot of people in this room. So just let me share my screen with you so that you can see my presentation. And let's start with this. So first of all, after saying thanks, I hope that you are all safe at home and healthy. And can you see my presentation now? And can you hear me? Yeah. Okay, perfect. So let's start with the topic of today. I'm going to talk about the uterine microbiome. This is my disclosure. I'm the research co-director at iGenomics Boston. And unless today I'm in Valencia, in Spain, due to this weird situation with the COVID and the restrictions to travel. And why talking about the microbiome? So, well, because we all are holobionts, and behind this very weird word that describes us, this means that we are all a cocktail of different types of microorganisms that inhabits our bodies. So we have our own cells, tissues, and organs that are of human origin. And this is completed with a load of microorganisms that makes the full body. And this full body needs this microbiota. This needs these microorganisms to perform very basic functions, very physiological functions, like for example, the digestion, the training of the immune system, and so on. So we have traditionally considered the microorganisms like something pathogenic or something that we always relate with disease. But in the last decades, we have learned that they are also good guys in our body. What about the urogenital tract microbiota? So we know from the majority of the studies done in the vagina of reproductive age women that the this niche is mainly composed of Lactobacillus species. But also we can found other type of bacteria, for example, Gardnerella, Bifidobacteria, Atopobium, and so on. And in turn, we have, there's a very complex cross-communication between the microbial cells and the human cells. So at the end, the microbiota can influence or can impact in different pathophysiological conditions in our body, and these conditions will also be related to the different microbiota that we will observe in the different niches. And this is a diagram that more or less represents these different interactions. What we know about the impact of an altered vaginal microbiota in infertility and obstetrical complications is that there are some bacteria that are direct causes of infertility. For example, Neisseria gonorrhea, Chlamydia trachomatis, and so on. And we also know that up to 40% of the patients that attend the clinics for IVF treatments present an abnormal vaginal microbiota, what we usually call bacterial vaginosis. And this bacterial vaginosis is responsible for increased risk of early miscarriage, late miscarriage, premature rupture of membranes, and also preterm labor, preterm birth. But what about the endometrial microbiota? So for those that work in human reproduction, the scenario that we like to study and where the action takes part is the endometrium, is the uterus where the embryo has to implant, not the vagina. So we started some years ago studying whether there is an endometrial microbiota. And this is a timeline that represents the different situations through the history about the sterility of the uterine cavity. So in the beginning of the 20th century, everybody thought that the uterus was sterile. Then the mid-20th century, people started to say in different publications, yes, there is an endometrial microbiota, or no, there's not. There's a lot of controversy. And thanks to the advances in next generation sequencing and technological advances from 2007 today, everybody that has been searching for an endometrial microbiota has found one. And this is mainly because we are now using molecular techniques in which we analyze the bacterial DNA instead of using the classical culture. We know that classical culture is not able to isolate some of the bacteria that grow in the reproductive tract, well, in the body in general, but also there is another complication about the another limitation of the endometrial microbiota, and this is because the endometrial microbiota is a low biomass microbiota. This means that the total amount of bacteria in the endometrium is approximately 10 to the 2 or 10 to the 4 times lower than in the vagina. So many of the times when you have a negative culture, is not because there's no bacteria in there. Sometimes it's because the amount of bacteria is under the limit of detection of the technique, and then we consider this negative. Based in studies that were using endometrial culture, we have learned that the isolation of common pathogens in the uterine cavity is correlated with lower implantation and pregnancy rates in IVF patients and increased miscarriage rates. So with all this background, we started to think about studying this endometrial microbiota at the molecular level and see whether there is a correlation between this endometrial microbiota and the success or failure in implantation. And this is the work that we published in American Journal in 2016. For this, to cover this objective, we started with patients with recurrent implantation failure. It was a pilot study with 35 patients in which they were going to undergo an endometrial receptivity analysis for the detection of their personalized window of implantation. So just before the endometrial biopsy, we collected an endometrial fluid in which we analyzed the microbiota. And then after the personalized embryo transfer, we follow the pregnancies so that at the end we could associate the results of the microbiota with the results of the live births from these patients. These are the results that we found in our setting, in our experimental setting. In this heat map, you can see any of the rows belongs to the most abundant, one of the most abundant bacteria found in the samples, each of the columns corresponds to the different patients that were participating in the study. And as you can see, there were some patients that had a higher abundance of Lactobacillus or Streptococcus or Bifidobacteria or Gardnerella, for example, in this set of patients here. So we ask ourselves whether we will be able to predict the reproductive outcome of these patients based in their bacterial composition in these endometrial fluids. To answer this question, we used two different mathematical models, and both of them gave us the same answer. And the answer was that the percentage of Lactobacillus in these patients was significant to predict the reproductive outcome with a cutoff value of 90%. So we classified our patients in those having more than 90% of Lactobacilli in their endometrial fluid samples, and we call this group Lactobacillus dominated microbiota, and those having less than 90% of Lactobacillus in their endometrial fluid samples, and we call this group non-Lactobacillus dominated microbiota. Then when we went back to the actual results of their treatments, what we saw is that in those patients with a non-Lactobacillus dominated microbiota, this means that the amount of Lactobacillus is below 90%, and of course, the amount of dysbiotic or pathogenic bacteria was higher. The implantation rates, pregnancy rates, ongoing pregnancy rates, and live birth rates per transfer were lower than in those patients having a higher amount of Lactobacillus. So it seems that the Lactobacillus really associates with reproductive outcomes in IVF patients. This result can be also been seen here in this graph, in which this bar in the left-hand side represents the average composition of bacteria in the woman that had a live birth. In the middle, you have the woman that had a miscarriage, and in the right-hand side, the woman that did not become pregnant after the embryo transfer, personalized embryo transfer. And as you can see, in the woman that had a live birth, the amount of Lactobacilli that is represented in blue is higher than in those having a miscarriage or no pregnancy. And in contrast, in those two groups, the amount of, for example, Gardnerella, Streptococcus, Veillonella, and other dysbiotic or pathogenic bacteria was higher. So this gave us the first information about this possible association between the microbiota and reproductive success. From this time, we have been doing a validation study, a prospective study, in which we have recruited 452 patients worldwide in 8 different countries. And our idea was to increase the variability, increase the sample size, and corroborate whether these results that we had from our pilot study are still valid. So we are now analyzing those data, and we are waiting for the last babies to be born, but we have analyzed all the samples in the lab, and we are starting to analyze the results, and hopefully the paper will be ready very soon, and I will be able to share the results with you in the next time that we meet. And from this time, we have been also working from another perspective, because we were interested not only in the abundance of the different bacteria and how many bacteria we have of each type, but also we were very interested in knowing what are these bacteria doing in the endometrium. So which are the functions of these bacteria in the endometrium? And to answer this question, I would like to show you a couple of clinical cases that we have published recently. The first one is this one that was published in the journal Pathogens at the end of the last year in 2019. And this is a case in which we have this patient that was presented to clinic with primary infertility and 1 previous IVF cycle. And the novel of this case, of this report, is that we were able to do a follow-up of 18 months of this patient, in which we have collected 6 different endometrial fluid samples throughout this time. This is novel because, as you may imagine, sampling the vagina is quite easy, but sampling the endometrium is not that easy. And mainly if you are working with women that are undergoing assisted reproduction, if you have to sample the endometrium in different months, this means that they are going to delay their treatments, and this is of course not the objective. So it is very, very difficult to sample, to have longitudinal studies in endometrial samples, and to my knowledge, this is the first one that analyzed the samples longitudinally. And well, as you can see here from these 6 samples that we collected over this time, we had 3 of the samples that were collected prior to a personalized embryo transfer. So this sample was collected prior to this personalized embryo transfer with a frozen euploid blastocyst, and this transfer ended up in an ectopic pregnancy. The second transfer also with a euploid blastocyst finally end up with a clinical miscarriage at the 9th week of gestation. And the last one, again with euploid blastocyst, also finished in a clinical miscarriage after 6 weeks of gestation. So also I would like to highlight that this patient had different antibiotic treatments between the samples, as you can see here in this gray color in the bottom of the bar. And I'm going to show you now which were the results of this microbiota at the different time points. So this is the composition of the microbiota in the different samples, and there is a common pathogen that is present in all of them, and this is Gardnerella in green color. And although we were treating the samples, the patients, with metronidazole and also with amoxicillin clavulanic, we were completely unable to get rid of this Gardnerella. So these were the results of the composition of the microbiota. But as I mentioned before, we were very interested in following also which are the functions that these bacteria are performing in the endometrium. So we collected in this time, the last sample that we collected in October 2018, and that is associated with the last miscarriage, we took the precaution to take this sample in the same cycle that the embryo transfer was performed. So only 7 days before the embryo transfer was performed, we collected first endometrial fluid, and then 7 days after we did the embryo transfer. With this design, what we were avoiding was possible fluctuations between the different cycles in the microbiota. So we are sure that the microbiota that we observed 7 days prior to embryo transfer was more or less what the embryo faced when entered the uterine cavity. When we move to the functional level, what we saw is that this is the same sample analyzed using only taxonomy or taxonomy plus functionality. What we saw is that in this case, although the abundance of the bacteria, the Gardnerella, was close to 20% when we only see the taxonomic data, when we go to the functionality, nearly 80% of the genes that were expressed at this time were expressed by Gardnerella vaginalis, which is one of the most fastidious pathogens of the reproductive tract in women. And when we analyze the set of genes that were expressed by these Gardnerella vaginalis, the majority of them were associated with virulent factors, with antibiotic resistance, biofilm formation, and other types of virulence factors. So it seems that this is impairing the environment of the uterine cavity. The other case that I would like to show is this one, in which we had a complete different situation. This is the case of a young woman. She was 28 years old when she came to the clinic, and she also had primary infertility with one previous IVF cycle that was failed. And well, she underwent this ICSI cycle with 6 euploid embryos that were vitrified. She had a first embryo transfer with 2 frozen euploid blastocysts, and after this transfer, the woman did not become pregnant. So because the embryo factor was discarded because she was doing PGTA in their embryos, the doctor decided to analyze her endometrial factor, and then she underwent this ERA test, and at this point is when she was started participating in the study. So we collected also the endometrial fluid for the endometrial microbiota study, and then after that, she had a personalized embryo transfer, again with 2 frozen euploid blastocysts. In this time, she was pregnant, but she had a spontaneous abortion at 8 weeks and 3 days of gestation. And of course, after that, she had accurate dates. We analyzed the product of conception, so we were able to see that indeed the embryo was euploid, because we had 46 XX results of fetal origin. And well, she had this treatment, this procedure, and she went back home to recover from the procedure. She spent the summer break, and after the summer break, she came back to the clinic just to have a checkup and see whether everything was okay and everything was ready to start a new cycle for embryo transfer. So at this point, the doctor took a second endometrial fluid sample to repeat the endometrial microbiota. And 2 weeks after this endometrial fluid sample, the patient contacted us to say that she was pregnant. She had achieved a pregnancy, a spontaneous pregnancy at home. And well, we were very surprised because, of course, she told us that when the doctor has collected this second embryo, sample, she was pregnant, 4-week pregnant. Of course, the doctor didn't know about this pregnancy. The patient didn't know about this pregnancy. So it was a big surprise for all of us. We stored these samples very carefully, and we were following the pregnancy until she delivered a healthy baby boy with no obstetrical or delivery complications, with an elective cesarean delivery. And after the baby was born, we asked her permission to analyze the samples and to publish the results. So I'm going to show you the comparison of the sample that was collected previous to the miscarriage at the sample that was collected when the patient was already pregnant, so during the early pregnancy. These are the results of the taxonomical composition. So this is the amount of lactobacilli that she presented in her endometrial microbiota before the transfer that ended up in a miscarriage. This was the percentage of lactobacilli that was in her endometrial fluid when she was already pregnant. And as you can see here, this is more than 90%. Also, when we went to analyze this more profoundly and we went to the species level, not only to the genera, what we found is that only functional bacteria that was present in the sample during pregnancy was Lactobacillus iners, while in the sample that was collected previous to miscarriage, there was a mix of different bacteria, but the Lactobacillus that was present there was Lactobacillus crispatus, not iners, which is, well, it was quite surprising for us. And going a little bit deeper in the functional analysis, what we saw is that there's a set of genes that is completely on during the miscarriage and off during the pregnancy, and the other way around. So the genes that were expressed in the sample previous to miscarriage correspond to different microorganisms. I don't know if you can see the legend here, but I can tell you that corresponds to, for example, Micrococcus, Lactobacillus crispatus, Pseudomonas, Enterobacter, and other types of bacteria. And they are associated with transposases and insertion elements, which means that this is represents like genomic instability. It's this type of genes. However, in those genes that were on during the pregnancy and were off during the miscarriage, we found all the genes that were expressed by Lactobacillus iners, as I said before. And the majority of the genes represented very basic functions, functions like metabolism, energy production, and some defense mechanisms. So the conclusions of our work is that the percentage of Lactobacillus together with the specific pathogens of the reproductive tract are significant variables to predict reproductive success. That these results expand the evaluation of the endometrial factor, not only at the morphological or the transcriptomic point of view, but also from the microbiological point of view. And also our observations lead up to propose new diagnostic tools and personalized therapeutic procedures through molecular microbiological evaluation. So this is our concept, the hypothesis that we have, and is that very simple. the better is your microbiota, the bigger your chances to have a successful pregnancy. And we are working in this concept, trying to develop this test to analyze the endometrial microbiota and the analysis of chronic endometritis. So just to finish, I would like to thank, this is my research group, so all the people working in the microbiota project, of course, Professor Carlos Simon, which is the boss and always support this type of research, so we are very grateful. All our collaborators, the iGenomics family worldwide, and of course, thank you to all of you for your interest and for your time today.
Thank you. Really great presentations, and we have a ton of questions. We're going to start out with a few questions before we do our next talk. So one of them, do you think the association with pregnancy relates to the decreased number of Lactobacillus or the increase of Gardnerella or Villanella or some other bacterium?
Yeah, this is a very good question. And from our pilot study, it's true that the only significant variable that came up was the percentage of Lactobacillus. But then after accumulated experience, working with a lot of samples and working in different environments and different practices, what we think, and this is only a hypothesis and we have to continue working on that and to really study this topic, we think that the role of Lactobacillus is not that important. So the role of Lactobacillus is to avoid the presence of pathogens that really can put the pregnancy into risk. So I think that because the sample size in the pilot study was quite small, for example, Gardnerella was not significant at that point, but maybe in our bigger study, the validation study, we will see that there are other pathogens that can be associated with, for example, clinical miscarriage or with biochemical pregnancy. We think that really the problem is not only to have Lactobacillus, but to not have pathogens.
Yeah, great presentation, Dr. Moreno. Thank you so much for that. We're going to do some more question and answers here at the end. We're going to go to our next speaker, Dr. Alicia Broussard. While she's pulling up her presentation, I will introduce her here. Her lecture today is on the sperm DNA fragmentation and its impact on IVF outcomes. Dr. Broussard has been an embryologist for quite some time now. Right now, she's an embryologist with Tomorrow Life Sciences. Today's presentation is a summary of her PhD research at EVMS, focusing on the effects of sperm DNA fragmentation on IVF outcomes and embryo genetic diagnosis. This research also focused on the best sperm processing techniques, sperm source, and for the selection of the best sperm with minimal DNA fragmentation. So it's a great pleasure to introduce to you Dr. Alicia Broussard.
Hi, everyone. Thank you for joining me today and giving me the opportunity to present some of my data for my dissertation work. So again, my disclosure is I'm the Director of Embryology at Tomorrow Life Sciences. When these studies were conducted, I was an embryologist at Midwest Fertility Specialists. I was also a PhD student at EVMS. The studies that I'm going to present are the collaboration with these organizations as well as with Reprosource. Reprosource did all the analysis for the DFI that I'm going to present to you today. So we all know that infertility is really complicated. It's often really difficult to determine the source of infertility. And also the female partner has far more diagnostic testing and previous research that's been done to determine her role. We still have a lot of gaps in knowledge about the role of the male and the sperm. So I wanted to really focus on the sperm aspect and how that might affect IVF. Over the years of working in the lab, I had noticed certain evidence that there might be more to do with the sperm than we were giving it credit for. So this is an example of the advanced semen analysis report. So this is a diagnostic test that it's a diagnostic test that has really changed how we can look at the health of our sperm. So in this diagnostic test, there are 3 values that are given. One is the DFI, which is DNA integrity. The other is oxidative stress index. That measures the damage from radicals. And then the last is high DNA stability. This measures a lack of decondensed DNA, which indicates immature sperm. So for all the sperm that was collected from my study, these 3 values were given for each sample. We also saw the invention of next-gen sequencing. We all know how powerful this platform is and how it's changed how we can diagnose our embryos. Not only can we measure aneuploidy and euploidy, but we can detect and measure mosaicism and segmental aneuploidies. Previous studies that I had looked at in the literature were not able to use this platform that were published at the time that I was developing my project. So then I started to think, if I could combine the use of these 2 powerful tests during my IVF cycles, could we get more information about the relationship with sperm and embryogenetic diagnosis? So for my study design, the first question was, are IVF outcomes, including embryogenetic diagnosis, correlated with DFI, either in the neat sample when it's collected or the sample when it's used at the time of insemination? Studies that I looked at in literature had really never looked at using a sperm sample on the day of, and they were, and I did not ever find a study that was published at the time where they actually used a sample after processing and compared it to any outcome. So for my study design, as again I mentioned, DFI, we need to look at it on the day of because it can vary from ejaculation to ejaculation. It can vary day to day, month to month, year to year. So I wanted to have the patient collect a sample. So they collected the sample by masturbation. I let it liquefy. And then after the semen analysis portion, froze a small aliquot. The sample was then processed per gradient protocol. And then after the gradient and wash, another small aliquot was frozen. The sample was then incubated till the time of IVF or ICSI. Following IVF or ICSI, the remaining sperm sample was frozen. We blinded the study by using random numbers and sending batches to ReproSource. The data was also not given to me until the study was complete to avoid any sort of bias. I also wanted to kind of get a double, by using this method here, I kind of did a little kind of double dipping in a way because I was able to look at the effects of both processing and incubation on sperm to see with previous studies that had shown that incubation might increase DNA fragmentation. So in our lab, we did the standard pretty much lab protocol for PGTA. We used Gardner grading system, and we only did day 5 and 6 transfers. The embryos were hatched on day 3 and moved to fresh media and corral dishes. And the embryos were graded. All of the sperm work I completed myself just to make sure it was consistently done for the study. But all the rest of the work as far as embryo grading, biopsies, et cetera, were a combination of myself and my lab mates. So for data collection, we looked at fertilization, good day 3, the number of good day 3 embryos, and that was relative to fertilization. The number of good quality blasts relative to fertilization. So this was defined as an A or B inner cell mass interfectoderm. And then we also looked at what we called the poor quality embryos, which had an A or B inner cell mass, but had a C interfectoderm, and those embryos were still biopsied. And then we also looked at the number of blastocysts that developed but were not suitable for biopsy. We looked at the rest of day 3 embryos, and then of course, the genetic diagnosis of the embryo and what day it was biopsied. We then also looked at the actual blastocysts that were biopsied on individual basis. So on each blastocyst, we looked to see what day they were biopsied, the age of the female or male, the sperm source, the method of insemination, the grade, the genetic diagnosis, and the sex of the embryo. For the male patients in our study, we looked at the sperm source. In the main study, we included surgical samples as well and frozen. We include the age of the patient, lifestyle factors such as smoking, alcohol consumption, BMI, and some other environmental factors. We also kept track of their semen analysis, and of course, change in the DFI, OSA, and high DNA sustainability between time points from neat to post-processing to at the time of insemination. So I obviously can't show you all that data here today. We don't have enough time, but I just wanted to point out the things that I thought were most relevant to my conclusions. So first of all, the DFI at the time of insemination is significantly correlated with the sperm's ability to fertilize the oocyte. I determined this with multiple different statistical methods. as well as patient level data and aggregate data. We looked at multiple cutoffs from the less than 15, greater than 15, less than 20, greater than 20, less than 30, greater than 30, and to look at the significance, and it stayed true. We also looked at standard versus ICSI, and it was also significant. The second point was the neat DFI at the time of insemination does not predict embryoploidy status. So this is, we looked at segmental aneuploidy, mosaicism, and obviously euploid and aneuploid. This agrees with other studies out there that use different platforms. There was no correlation with blastocyst development or embryo quality in DFI, in either the neat sample or the sample at the time of insemination. This was a little surprising, just because other studies consistently out there have shown that there is. Statin insemination resulted in a significantly higher number of blastocysts and euploid embryos compared to ICSI. Preparation by gradient and wash did improve DFI, but not in all of our patients. We overall saw about a 40% decrease in DFI by using the gradient, but we had around 16% of our men whose DFI actually went up during processing. And we did not end up seeing a difference in pregnancy rates when a euploid embryo was correlated with DFI. So then I went on to the next portion of my work and thought, how can we get a better sperm? If we know, at least from our data, that it's affecting fertilization, then we're affecting the number of embryos that we're starting with to be in the very beginning of the cycle. So the first part is a little study I did. So this was 30 random semen analysis patients. Our andrologist at Midwest Fertility would complete his semen analysis, de-identify the patient, and then hand it over to me. These were all random. It would be, we're gonna do 5 this afternoon, let's go grab 5 random patients. So these were all random. They were all across the spectrum, from very, very low counts and motilities to, very normal. So the sample was already, again, had already been analyzed, so it was liquefied. So he passed it off to me, and I divided the sample 3 ways. So actually I took so I divided the sample, I took an aliquot off the neat sample for the neat sample, and then I divided the rest of the neat sample between isolate, sperm grad, and the zymote separation device. So I went ahead and did these per protocol per their instructions, and then at the end, the zymote device, I pulled the sperm out of the outport and froze that. For the sperm grad and the isolate both, following their processing, I took off an aliquot of that, and then the rest of their sample I performed a swim up with. So following an hour of swim up, an aliquot was taken from the very top of the media. This is what we determined with the DFI changes with those processing methods. So again, we were very impressed with the zymote device. I hope I see a lot more research out there with that. We also analyzed the data, comparing each of the methods against the neat and each of them against each other. And the good news is that we did see all the methods were effective versus the neat versus the neat sample to reduce DNA fragmentation. So many clinics I know have gone to patients with failed previous cycles and high DNA fragmentation, considering doing a taster with. So we wanted to find out what would happen with those patients. So this is only 3 patients, I realized, but I still thought that the data was interesting. So these patients collected a sample right before surgery, and the sample was taken to the lab and was processed just like we processed for IVF. It was then held in the incubator. The surgical samples were obtained, they were processed, and they were, held until it was time for ICSI. Once ICSI was complete, both of those sperm samples were frozen. So they were both really exposed to incubation and such for the same amount of time. And so what we saw here that I think is important to note, that yes, they all did have a high DFI at the time of the morning of their surgery. However, all 3 of these were processed down to a low amount, and they stayed, and this is after incubation. So this is after processing and incubation, and they all ended up being lower than the testicular sample that was obtained. So then we thought, let's just look at some of our when we have leftover sperm from a case of our surgical patients, or if we have multiple sources, let's see what we find of these samples. So again, I'm just going to focus on DFI for the sake of time. But what we saw here is a couple of things that I thought were interesting. So, so patients with both types of samples, epididymal versus testicular, the epididymal was much higher. and I was surprised at how high some of these were. It made me think back to my days when, you know, we used to just, for our post-vasectomy or our vasectomy patients, we would just go in and aspirate some epididymal fluid. We also had DFI was much higher than I expected in some of these the testicular samples. And I also thought it was interesting to note that the vasectomy patients overall compared to the patients with azoospermia or obstruction, they actually were consistently lower. Again, I would love to see more work on this. I'm sure other people might be doing something similar, but at the time, this was, you know, pretty novel. Oops, I think I skipped a slide. There we go. So follow up for this work. Again, as I mentioned, some of my data does not match some of the other publications out there, which, you know, I think it would be interesting to go back and do some more of the study, but can maybe do some controls. So for instance, the speculation that healthy oocytes can repair DNA fragmentation, it would be nice to have a healthy population of oocyte donors in a large quantity that we could look at different levels of DNA fragmentation and the outcome. It's a little bit difficult for research sometimes because our patients that we see in the lab are already infertile couples for the most part. So again, some of my results that did not turn out, like for instance, blastulation being lower in men with high DNA fragmentation. It could be because of our patient population. They were already infertile patients. They were presenting for PGTA, which meant that they probably already had a failed cycle. I would also like to see, again, more studies for DFI on the day of oocyte retrieval. I think it's really important to use the sperm sample that you're actually going to use for the insemination, knowing that it does change greatly over time. And then again, more studies just about sperm in general. We saw that regular inseminated embryos had higher blastulation and higher euploidy rates. So if we have better ways of knowing we have really healthy sperm and that we have the best culture conditions for fertilization, we could probably reduce ICSI and that might improve our outcomes. And I always think it's important to repeat studies in other laboratories and on large magnitudes because different populations of patients change per area, as well as other laboratory conditions are different. I just want to thank all the people who helped me work on this project, from Dr. Leader, who owns Reprosource, and then all of my, Dr. Edney is my lab director at Midwest, all the doctors, Dr. Culver, Reuter, Bob, Will, and other Will, for all their hard work. Dr. Torado, who unfortunately passed last year, but she did all of the analysis for the DNA fragmentation. Dr. Hinbeidu, who taught me statistics, and Dr. Kovac that I worked with to collect the surgical samples, Dr. Russell, who helped me with all my IRB, and then of course my awesome lab mates, Tricia, Alice, and Jen, and my EBMS class, and all those patients that participated. Thank you.
Excellent presentation, Alicia. Thank you so much. Thank you to both the speakers for coming out today and presenting their data to us. We'll jump into some question and answers to both speakers. I know that there's been a number of questions to Alicia. What method were you using for measuring DNA fragmentation?
We were using, as in the SCSA method, they call it SDFA at Reprosource, but it's the same method with the exposing it to the low pH, the acridine orange, and then flow cytometry to measure. And then the OSA is actually a test that Dr. Torado developed herself that's been validated. That's actually something that only Reprosource offers.
Another question to Alicia. What do you think is the best method for sperm DFI evaluation?
Well, I like the SCS. You're asking which test, I guess. I mean, so the tunnel is more of a direct test and the SCSA is more of an indirect test. But I think there are studies that show that they don't necessarily always line up, but I think that either one is good and would give you enough of an answer as if there was an issue with DNA fragmentation. I think both are definitely sensitive enough and both are great tests.
Do you recommend DFI in every patient?
I would say yeah, because honestly, first of all, we know from lots of studies that DFI can greatly, can be a real harm as far as natural insemination, getting pregnant, pregnancy loss, that type of stuff. So I think if we especially if we know more about processing and how that translates over to a healthier cycle, then patients could all be tested. And the only thing that's tricky is I think you need to know the sample on the day of. So I would say testing is important, but I think more important is finding out ways in your laboratory to reduce it.
Good. To Dr. Moreno, we have a question here that says, is the microbiome related with endometrial metabolic secretomes?
With secretome?
Just, is the microbiome related with endometrial metabolic secretomes? Like, is this naturally occurring? My question to go with that would be like, are there things in our diet that we could use just naturally for our lactobacillus? Because that seems to be the key one to have.
Yeah, absolutely. This is not part of our research, but there's a lot of research in the gut microbiome. And of course, the substances that the bacteria can collect from the gut and then they can process and metabolize and secrete are absolutely related with different pathologies. We don't have this knowledge from the reproductive tract, but there is like kind of a hypothesis that in many organs, the microbiota is orchestrating other physiological functions. And also there are connections between the microbiota in different body niches. For example, there's studies that shows that the microbiota in the intestine is related with the gut-brain axis. So some conditions that happen, for example, depression or anxiety, are orchestrated by the intestinal microbiota. We don't have the knowledge now about if the endometrial microbiota is also connected somehow. There's some papers that points to all the organs that are regulated by estrogens may have this type of regulation and the connection with the different body niches and the different organs. But there's a very interesting field of research for the future.
Excellent.
Now, what about sampling time? Is this a luteal phase test? Is the tissue collection time important?
Yeah, there's in our case, we always collect the samples at the same time during the window of implantation. So because there's some studies that are performed in the vagina that says that the microbiota may be hormonally regulated across the menstrual cycle because of the estrogens and the progesterone. So because our interest is in the window of implantation and the moment in which the implantation takes place, we always collect the samples in this time period so that we can, let's say, mimic the needs that is going to be present during addition and implantation of the embryo.
Alicia, coming from Michael Reed, he asks, does a high DNA fragmentation index in the ejaculate indicate a generalized problem with a sperm population, or does it only reflect on issues with the sperm affected by fragmentation? In other words, does processing and reduction of DFI actually change the outcome compared to unselected sperm?
I would say that that's still really unknown. There's a lot of people that would say they think that whatever the DNA fragmentation was in the sample, that's just kind of indicative of the whole health of the sample. I would say that we still don't really know for sure. I think also it varies. I mean, we might have a patient that just chronically has high DNA fragmentation, and you come to find out that there's other factors going on. Like that's been, like in my study, I found that there was a link between people who drink every day and DNA fragmentation. So I've always thought that the DNA fragmentation could maybe not be the cause of poor IVF outcomes and pregnancy loss, but might be indicative of something else. The high DNA stain ability that I mentioned in my assay, that indicates in immature sperm, which it measures the histone retention. So when we had patients that had high DNA fragmentation, they also frequently had high histone retention, high histone retention. So that means that instead of having, 2% histones remain in their sperm DNA, they have 50%. And so to me, sperm DNA fragmentation and something like histone retention might be something that, you know, they're kind of maybe a symptom. But I think that in a population, you can have healthy sperm and poor sperm.
Okay. In your cases, were they all ICSI, or did you do a mix of conventional and ICSI?
Yeah, we did a mix. So a lot of our patients will do like 50-50, or they'll do 3 ICSI and do the rest in regular insemination. And then there were some that were 100% either way. So we would keep track. They're kept separate, whether they're ICSI or regular insemination. So I was able to know every embryo for biopsy, where it was derived from.
Yeah, in the early DFI days, we did a small study with conventional INSEM versus ICSI. And just like a lot of the initial studies showed no pregnancies with high DNA fragmentation, but when we did ICSI, it seemed to normalize. So just curious what your outcome was with that.
So if the theory is that you can reduce your DNA fragmentation and you have healthy sperm in a population, then my theory on that is if you're doing ICSI, you're going through your hand selecting a sperm. So usually so in my study and other studies, we did some correlations with like morphology. So if you're able to go through and handpick your sperm, then there's a high DNA fragmentation in your sample, maybe you're just more likely to pick the pretty sperm that's the best developed.
Yeah. Cool.
Alicia, can you explain the mechanism by which the DNA fragmentation reduction in the zyma?
So the Zymote device is a little chip that basically it has an import and export. So it's a circle that's basically, I don't know how best to describe it, but what you do is you put culture media into it and to kind of like prime it, and then you load your sperm slowly onto one side with a syringe, and then you wait for half an hour with incubation, and then you go to the out port and you syringe out I took about 0.4 ml and froze that.
Okay.
So the chip, you can put a maximum of 850 microliters of semen on it.
Okay.
They just stick it in the incubator.
Another quick question. Would you recommend antioxidant therapy for abnormal DFI?
I'm not sure. When I was working on my lit review, I read quite a bit about it, and I still don't think we have enough evidence that it works or which one would work.
Okay.
Yeah. Dr. Moreno, there's been a couple questions on sampling. How are you able to take the sample from the uterus without contaminating it with vaginal microbiome?
Yeah, so avoiding contamination in the vagina is quite easy. We collect the samples with a embryo transfer catheter. So we, of course, we always clean the vagina, and we are very careful to enter into the uterine cavity without touching the vaginal walls. In any case, we use a double lumen catheter. So we only expose the inner catheter when we are already inside the uterine cavity. So avoiding vaginal contamination is more or less easy. I would say that it's a little bit more difficult to avoid cervical contamination. But in any case, there's a paper that was published in 2017 in which they analyzed the reproductive tract at different sections. So 3 points in the vagina, then the cervix, the uterus, the fallopian tubes. And they say that the microbiota in the cervix and in the endometrium was very similar. So what we consider is that if there is any potential contamination with the endocervical microbiota, this may not affect, because the close proximity of the two niches and the close proximity of the two microbiotas. So it seems that this is the cleanest way to do it. Of course, there are many other, for example, the paper that I just mentioned, they work with hysterectomy pieces, but of course, working in reproduction, there's no other way to sample the vagina, the endometrium that's going through the vagina and the cervix. So we try to do it in the best conditions.
Spectacular. We, early in my career, we used to blame our miscarriages a lot on genetics. And now we're really good at looking at the genetics, knowing we have the right embryo, or I like to put it in the context of seed and soil. So I've always liked the microbiome studies based on the idea of, like, if we have a good seed, we have a genetic report to tell us that. And then we have the soil. Now maybe there's something in the soil that's just either missing or something that's not right in the soil. Is there something in that and that we can test at the time of, say, retrieval? Do we do one of these samplings and then over the 5 or 6 day growth period know if the endometrium is going to be receptive based on the microbiome? Is this something we could do clinically today?
We haven't done that. we really don't know. We really don't know. But I agree with you in this, and in fact, the way in which we started these studies is because, of course, we know that in women that have that perform PGTA in their embryos and they select the best embryo, and they also do like endometrial receptivity analysis, and they know exactly their perfect window of implantation. Even if you have the best embryo and you have the best endometrium ever, there's many women that still don't get pregnant. So there are many other factors, and microbiome can be one of them. But of course, there may be others also playing an important role in implantation. So we consider that and this is mainly my point of view, because of course this is the work that I perform, but I always think that if the bacteria, not only the bacteria, also viruses, also yeast or fungi are present in our bodies, and they are a lot of cells growing in our bodies together with us, this may be for something. I mean, it's not just by chance. They are doing something, they are playing roles. I don't know if this is cause or effect, but of course they are doing something and it deserves further investigation. And I consider that this point that you raise is very interesting.
Do you think the hormones from the stem cycles would affect the microbiome at all?
Sorry, can you repeat?
Do you think that the hormones during the stem cycles for IVF can be affected by microbiome?
Yeah, I think so. And there's the studies in the endometrium are there's very few studies in endometrium, but there's studies in the vagina. And of course, the endometrium, the way in which the endometrium is colonized is by the ascension of the bacteria from the vagina. And it is, I mean, you can see that during the menstrual cycle, there are periods in which the microbiota is more stable or it's more variable, and you have more, you are more prone to have pathogens or to have lactobacilli. So hormones are really important for the composition of the microbiota in the reproductive tract, not only in the endometrium, but also in the vagina. And I think that is clearly demonstrated. There's one thing that many people ask me, and this is something that we haven't achieved yet, and is to see whether the hormones that we gave to the patients for ovarian stimulation, or what is the impact of this, or if it's better to, let's say, work with natural cycles or stimulated cycles or hormone replacement cycles. But this is, there's thousands of experiments and studies to perform.
Is there any probiotics that you would recommend to enhance endometrial lactobacilli?
This is a big question. I mean, there is no clear evidence on the role of the probiotics in the endometrium. There is some evidence that they act in the vagina, but so far there is no endometrial probiotics. So the closest way to deliver these probiotics to the uterine cavity is leave them in the vagina and hope that they will go up and colonize the endometrium. There's no many evidences about this, and there's no randomized clinical trials comparing patients that have been treated versus placebo. But this is the opinion of many of the doctors that we work with, and is that at least they won't harm the patient. So I know that after, for example, after treating with antibiotics, many doctors are asking us to give them probiotics, because the worst that it can happen is that they don't do nothing. And if they can go up and colonize, so perfect.
Great point. We actually say we can treat a lot of our illnesses through nutrition. So what we eat probably would affect it as well. But what about antibiotics? Should we avoid antibiotics? Well, I know a lot of programs will give antibiotics after retrieval, and that could be totally killing.
After retrieval and even before embryo transfer, there are some clinics that are giving antibiotics to the patients in every single stage of the treatment. I mean, I think that this will depend on the type of antibiotic, the duration of antibiotic. Not all the antibiotics kill lactobacillus, not all the antibiotics kill all the bacteria. So, of course, I think that we are living in a time in which we are abusing antibiotics, not only for reproduction, everywhere. We are all the time abusing the use of antibiotics. I think that now the procedures in assisted reproduction are quite safe, so maybe the use of antibiotics is not that necessary, and we should try to minimize it. But the effect on the microbiota will depend on the type of antibiotic, the duration, and also the susceptibility of or the community that this woman presents, because not all the antibiotics are going to perform the same effect on the microbial composition of the woman.
Great.
There's a question from Kimball. The molecular methods for determining microbiota are very sensitive. Were you able to determine quantity?
The molecular methods that we are using, it depends in which molecular methods you use. For example, if you are using NGS, you cannot determine the absolute abundance of the microbiota. You just can have like the percentages between them, the relations in between them, but not the global amount. For example, if you use qPCR, you can have your own controls and your standard curves, so you know exactly how many copies of the genes that you are analyzing you have in the samples. So it depends on every technique has pros and cons. So NGS is very sensitive. You can analyze all the samples, all the bacteria at the same time, but the limitation is that you cannot infer the amount of bacteria that you are quantifying. You cannot quantify. If you use qPCR, you have to attain to a panel of bacteria, but you can quantify them. In the last years, there's some techniques that you can apply to NGS, in which running like standard microbial compositions, you can infer the numbers of bacteria that you have in the sample. But this is not a direct quantification. It's just comparing what the input that you mix with your sample with the output that you recover from the NGS. So it's an indirect quantification.
So Alicia, I know we're getting towards the end of our time. There was a question like, does the egg have the capacity or the machinery to repair the sperm DNA if the DNA was high, DNA fragmentation was high? The idea with donor eggs would, even if it was high, would it have its own ability to repair that fragmentation?
So in sperm DNA fragmentation reviews that I've read, there have been studies that have been mentioned where they think that's the case. Although I think it's kind of tricky to figure out for sure how that happens. So I don't really know for sure. I would say that it's a really hard question to ask, especially I think that human oocytes and sperm are so different than other species. So for instance, back to the histones, mice never really have that issue with their sperm where they maintain those histones. So I think even using animal studies to try to determine maybe how the oocyte, repackages the sperm DNA and repairs it, you can't really do that ethically in humans. So I think it's hard to know. You'd probably have to do some extensive staining. I'm not really even sure how you would measure that mechanism at this point.
Yeah, that's the fun thing about science, right? Trying to ask those questions.
Yeah, there's a question, the second ejaculate is thought to have a lower DFI. What's your opinion on that from Iman?
I think it depends on the patient. I mean, I think it's possible if the patient has, I mean, we were having patients that, like there was a patient, for instance, we had in our lab, and I didn't put this in here, but we had a patient that had high DNA fragmentation. The first thing they did was they tried a TASA. When we found out later, his TASA was still like, I don't even know, 50% fragmented. So then he tried the whole protocol of like 3 days of ejaculations in a row and then take a day off. And so we ended up doing some testing and his DNA fragmentation never went down. Now another person that say is completely normal and he only has high fragmentation because he hasn't ejaculated in 2 weeks, then it's possible maybe that once he ejaculates and the epididymis is, that portion of the epididymis is kind of cleared out, that it's possible. But I don't know if it could be just one ejaculation.
Okay, thank you.
So looks like we're getting about to our time limit here. Going to wrap it up. The speakers today, we couldn't get to all of our questions. We're going to send these questions to the speakers and have them put some responses to those questions, and it'll be posted on the website along with this presentation. I'd like to thank CRB for allowing us to do this. If you are certified through the CRB, they will be approving credits for this for 1 hour. And there's going to be a follow-up CRB session here in a few weeks with some of our abstract presentations we've had submitted. I'd like to thank our panelists who are also part of the planning committee. Eva is one of the planning people, and I thank you for her for joining me and moderating this session today.
Thank you for having me.
Thank you. Thank you very much for your interest and for inviting me.
Thank you.